Hello, my name is Joseph Trackman, and tonight we're going to be talking about autism and, in particular, understanding autism. I would just like to make a personal note that today is the anniversary of the massacre of my grandfather's village in Poland, and a very special day for our families. And I hope that this presentation will be very special and help your families as well. So just very basically, we're going to talk about the history of autism, the diagnosis, and the treatment. I first became interested in autism in 1970 when I was a graduate student at Johns Hopkins University, and it was at Johns Hopkins University that Dr. Leo Kanner first described autism in 1943. I had the good fortune to be a student of Professor, well, I was a student of his students, of Professor Donald Baer at the University of Kansas at Lawrence, and he developed the ABA method for autistic children. So I didn't learn firsthand, but very close secondhand. From 1974 to 1990, I was on the board of directors of a school for autistic children that was courageously started by 6 sets of parents whose children had been at the Willowbrook State School. And you may know that is how Geraldo Rivera became famous with his expose of Willowbrook State School. And by divine providence, when I was teaching at the College of Staten Island, the grounds were on the Willowbrook State School. So I taught autism in the courses there. And since coming to Seattle, I've served on the board of directors during the founding of the Academy for Precision Learning and have published papers on autism. See, this paper I published in 2008, and since that time, our view of autism has changed quite a bit, and so we're going to discuss the changes and the implications of the changes since writing this article. And as you can see here, 2011, the article was #1 in the domain of autism 3 years later. So it was a very successful article. So here's a brief history of autism. Unusual behavior by children was known as childhood schizophrenia. As mentioned before, in 1943, Dr. Leo Kanner used the term autism. 1 year later, in Germany, Dr. Hans Asperger described a milder form of autism. His article wasn't discovered till around 1990, and that's when Asperger's syndrome started to become popular. It was in a German publication, which I read. I could read German, and that's what he described. Now, in 1978, the diagnostic criteria were further relaxed by Reuter, and now the diagnosis has evolved to autistic spectrum disorder. So I took this image from the internet, and why? Because there's so much confusion about autism, we can take a look at this and see why there's so much confusion. We have 12 different areas or categories or descriptions of people that comprise autistic spectrum disorder. Now, scientifically, you can't say that unless you hold 11 of them constant and change one. Or else you have what we call confounding variables. And when you have confounding variables, your conclusion is invalid. If your initial premise is wrong, your conclusion is going to be wrong as well. Secondly, if we even assume that these were valid, if you take all the combinations and permutations of the 12, you have a formula 2 to the n-1 power, which would be 2 to the 11th. And that's over 1,000 different possibilities, and we're going to discuss that a little later. So you can see how much confusion there is right now. Okay. The CDC report last year shows the prevalence of autism increasing from 1 in 10,000 to 1 in 59. And one of the reasons, in my opinion, is because of the relaxed criteria, but we're going to discuss other possibilities as well. Okay, so here's some questions that you probably have on your mind. If not, we'll put them in your mind and we'll answer them at the end. So, are the changes in the diagnostic criteria responsible for the reported higher prevalence of autism? Is glutamate a brain chemical transmitter related to autism? Glutamate is related to glyphosate, which is in Roundup and Agent Orange. GMO is related to the MTHFR mutation, which is also on YouTube, to vaccines and mercury. One question, yes.
Yes, Dr. Trackman. So I know that Agent Orange is related significantly in the historical records as related to like the Vietnam War. And Roundup has been related. Roundup is the pesticide, right? It's that the weed killer, that sort of thing. And then the GMO, the genetically modified organisms, I think. I know there's been a big push for non-GMO. Is there evidence of
In the next few slides, I'm going to discuss the relationship among all these different chemicals. Okay. Now, another question is, parents will ask, we have 2 children. One has been diagnosed as having a spectrum disorder, and the other excels in everything. Is there an explanation for this? So we're going to explain that also. And the last is there a genetic component related to autism? In the 2008 article, none of this was known. The Genome Project had not been elaborated as it is today. So this is a good question, and we hope to have a good answer. Now, this is very, very interesting. Synesthesia. Synesthesia is when a sensory input to the eye, for example, results in a person hearing a sound. It's been reported that only 1% of the general population have synesthesia, but 15% who have been diagnosed with autism have it. And this topic of synesthesia was made very popular, as did autistic savants with the 1988 movie Rain Man. Now, go to the diagnosis. There's many, many different theories about the diagnosis and the etiology of autism. And you can see here the different factors involved. Presently, the MTHFR mutation, Fragile X syndrome, and other genetic mutations have been included as possible etiologies. Now, we have a question. Hold on.
Can you explain really quick, super high level, what the MTHFR mutation is, just so people understand?
So once again, I'll give you a quick explanation of the MTHFR mutation, but I suggest you go to my YouTube channel and click on the epigenetics presentation that I gave a few weeks ago. But MTHFR stands for methylene tetrahydrofolate reductase. Now, say that 10 times fast, and then you'll have a good sleep. About 45% of the U.S. population have this mutation, and the mutation is of an enzyme that converts vitamin B9 folic acid to the usable form in the body, methylfolate. And as you're going to see in a slide, a future slide, why this is so important. Now, I have a caution here. In my opinion, there's two different categories of autism. There's the classic autism described by Kanner, which is severe behavior such as hair pulling, arm biting, head banging, rocking, echolalia, these different things, as opposed to spectrum, which, as you saw on the previous slide, anybody could have it because the criteria are so lenient. Now, this is also very interesting that there's a reflex, just like if someone goes to hit you in the face, your eye's going to blink. That's a reflex. It's something that you don't have control over. Okay. If you take a spinning drum that has stripes on it and you rotate it in front of someone's eyes, their eyes are going to oscillate back and forth. That's called optokinetic nystagmus. Now, if you're from New York City like me, you could observe that as the subway cars go past and the lights go on and off. I would stand on the platform waiting for the train and I would watch people's optokinetic nystagmus. I didn't get arrested, so I guess it was okay. Okay. People with the classic autism do not respond to the spinning drum. So there's something going on chemically or neurologically with them that differentiates them from the general population. So what I would like to do is present a unified theory that's going to put together all the different factors that we know about, including the chemicals and the mutations, and see if we can come up with something that makes it a little more understandable than it is today. So let's explore some of these relationships. Okay. So I don't expect you to remember, we're not giving a quiz at the end of the talk, so don't get nervous. But what I want to show is that glutamate, which is a brain transmitter, is a major component of glutathione. And glutathione and glutamate have been reported in the literature as being related to autism and to the excuse me, the mutations related to glutamate. Okay. Now, to answer Brian's question about the MTHFR. So here is the methylfolate, okay? Now, each cell in the body has this methylation cycle, whether it's a hair cell, a skin cell, muscle cell, heart cell, brain cell, cells in the eye. Every cell has its own individual methylation cycle. ATP is from the mitochondria, so the methylation cycles related to the mitochondria. Here's cysteine, which has been reported to be related to autism, and here's glutathione. So we start to get a picture now that biochemically everything is related, and I haven't seen anyone put all of this together in any of the literature that I've been reading. Okay, so here is the bad chemical, glyphosate, and you could, if you remember what the chemicals look like, it looks very similar. So glyphosate interferes with the DNA of whatever it's being assaulted, whatever is being assaulted. Okay, so once again, here's glutathione, okay, and here's glyphosate. So you can see a similarity, and this is why it's so effective, because it matches the natural chemical and goes in and disturbs the DNA. So here's this report. It documents how glyphosate interferes with the GMO, the genetically modified soybeans, and the Roundup is glyphosate. Now, you might ask, well, if glutamate's so important, how are we going to get our glutamate? Okay, so these are the foods that are rich in glutamate. Okay. So here I try to make a diagram or a chart showing the relationship between the different chemicals and mutations related to autism. So in MTHFR, as we just saw, it's homocysteine and cysteine. We're going to discuss this in a little while about a brain structure called the hypothalamus, and one component of that, the lateral hypothalamus, produces glutamate and another brain chemical called vasopressin. Mercury, we've all read about mercury in vaccines. Arginine and GABA also have been reported to be related to autism. GABA is gamma aminobutyric acid, one of the brain transmitters. After this, you may see this familiar as in some soft drinks that you shouldn't be drinking. And here's GMO, glyphosate, and this. Okay, so now we'll go to the next slide. So here, all I did is transpose glutamate for autism, and they're all related. They're all interrelated to each other. Now we're going to take a look at some of the basic genetic connections involving the MTHFR, the glutamate mutations, Fragile X syndrome, mercury mutations, and some others. This is a very interesting and informative article about epigenetics and autism. So if anyone is particularly interested in the topic, I would suggest starting with this article. Is very informative. Okay. And once again, if there's other questions about genetics, to go to my YouTube channel and click on the Epigenetics video. Okay. Now, we have here listed 1, 2, 3, 4, 5, 7 different on this side, 7 on. So we have 14 different mutations related to glutamate, adding to the confusion. Glutamate's been related to autism, but glutamate has these different mutations, not just one, but many. The two main ones are in bold print. Now, there are some mercury mutations as well, also adding to the complications and confusions which we're going to discuss in a little while. Okay. Now here's the hypothalamus. The hypothalamus is geographically in the center of the brain. So if we go in the center this way, and front to back, we'll hit the hypothalamus. Why is it there? Because it's the the hypothalamus is geographically in the center of the brain. So if we go to the center here, and the center here, and the center here, we will come to the hypothalamus. So why is it in such a deep place? Because it's essential for life. It controls all our autonomic nervous system functions. And one part of the hypothalamus, the lateral hypothalamus, is the one that produces glutamate and vasopressin. And the hypothalamus is small. It's only about the size of the top of my thumb. That's all. Okay. So here are some of the functions that the hypothalamus regulates. So you could see it regulates almost everything. It regulates the pituitary gland and the pineal gland and the retina and blood pressure, eye pressure, nitric oxide, the immune system, the emotions, memory, all these heart rate, peptides in the brain, body temperature, all these different things. So this gives you a sense that somehow with the glutamate production, if there's something wrong with it, it's also going to affect something else in the hypothalamus. Why? Because the hypothalamus is the head ganglia, the head nerve center for the sympathetic nervous system. And why is it called sympathetic? Because what happens in one part tends to happen in another. Okay. Now, here's another caution. One of the things that I'm very against is labeling anybody about their behavior. It's very, very dangerous. Why is it dangerous? Because it leads to self-fulfilling prophecy. So I was talking to one of my colleagues in the veterans program here at the WeWorks, and I thank the WeWork facility for allowing us to use the facility. And he has a son, and I can't get him to do anything. And every time I ask him to do something, if he doesn't want to do it, he just says, I can't do it, I have ADD. Very dangerous. Or they have spectrum disorder. So we know from years of scientific research that there's two very important effects. One is called the Hawthorne effect, and this is the student's expectation. If the first day of class, the teacher comes into the classroom and tells the class, the students, you are the smartest in the whole school. I'm so proud of you, and you're gonna do so great. And there's actually was a movie made, I don't remember, maybe in the 80s. It's called Stand Up, something like that. And it was about a teacher who was assigned to a class in a school in California that was full of not good students. But he told them that they were the best, and you know what? They ended up to have the highest scores on the statewide test. Now, there's also the teacher's expectations, which if they tell the teacher, this class, forget about it, they're never going to learn anything, what's going to happen? Teacher's not going to try. So labeling is very, very dangerous because we all strive to help each other and make each other the best that we can. By labeling, we destroy that.
Really quick, I just think that has, I think that goes beyond autism spectrum disorder. I think that goes with anything, whether it's like cerebral palsy or depression or like anything like that. Like, like when you you know, when you label something, you diminish it. But you can
Even
also limit
even in industry, if you tell your workers, "Wow, you guys did such a you're the best workers I ever had," they're going to be the best workers.
Yeah. It's all about that positive or negative reinforcement. If you're being positive about it in the reinforcement, they're going to be positive about it. If you're being negative about it, then you're going to have negative effects too, right? So regardless of whether it's autism or anything else, it's going to have that positive or negative effect. Am I right?
100% correct. So you're 100% correct, and that's why I'm so against labeling. So now that we've gotten a lot of bad news about all the problems, what are we going to do about it? So here are some of the treatments for autism and autistic spectrum disorder. Okay. So once again, this is very brief, and it's elaborated more in the epigenetics video, but there are supplements to compensate for the methylfolate mutations. And when prescribed properly, they're very, very effective. Now, one of the consequences of the MTHFR mutation, particularly in children, is low muscle tone. And because they have low muscle tone, their general motor development is faulty, and their vision development is faulty, and their eye-hand coordination is poor. So these children are going to have a variety of vision problems. I know you can't read it, but they're going to have trouble focusing. They're going to have trouble converging their eyes. They're going to have trouble maintaining visual attention. They're going to have trouble obtaining space information from their eyes. When these children come into my office, they're touching everything. So by the time they get from the office door to the examining chair, I already know what's going on, because they're touching everything all along the way. So these children have low muscle tone and a variety of motor problems and vision problems, and we get them methylated. So we go through the regimen to get them methylated, and we give them vision therapy, which we're going to talk about, and also recommend that they have physical and occupational therapy to build up their strength. So the last presentation was in 2013. In 2016, I presented this at the mitochondrial conference here in Seattle, and it's just relating the similarities between the methylation mutation and problems that people have with mitochondrial function. Once again, all these things are all interrelated with each other. They're not isolated problems. Okay. Now this is amazing. This was in the New York Times in 2007. And what was the article? Not autistic or hyperactive, just seeing double at times. So here, this is 12 years ago, almost 12 years ago, the New York Times reported, and how many millions of people read the New York Times, and saying, no, don't call people autistic, don't call them hyperactive, have their eyes examined, because they're seeing double. But apparently people forgot about it. So we're going to take a little vacation from autism, and we're going to go to the eye, something that I'm a little familiar with, since it's the only thing I've been doing for the last 50 years. Okay, so inside the eye, there's a lens, a focusing lens, and surrounding the lens is a ring muscle that when it contracts, the lens gets fatter and we see up close, and when the muscle opens up, the lens gets thinner and we see far away. And this is controlled by the autonomic nervous system, and a lot of the control is from the hypothalamus. So once again, we see this interrelationship among all these different apparent disparate variables, entities. Okay. So we're going to talk a little in a little while about the zone TRAC, which is one of my inventions that trains the ciliary muscle and helps improve concentration as well. Okay. Now, in the New York Times article, they talk about this vision disorder called convergence insufficiency. Okay, it's not about up in the north of Seattle where they have the convergent area where they have all the bad weather. There's not that convergence. It's that the eyes do not look at the object that they want to. So, for example, someone's looking at this book, but the eyes are pointing over there. So this is called convergence insufficiency. And what's the consequence of that? That they'll either see double, or they'll see almost double. They'll see a shadow image, or they'll just see one. The brain will turn off one eye to avoid the double vision. Okay. Another problem with the low muscle tone is that some children will have one eye that turns in or turned out, and the name for an eye turn is strabismus. When I first came home from optometry college, and my parents wanted to know, well, how they were spending their money. So what did you learn? I said, I learned strabismus. And they thought it was a bad word, so I had to explain to them that I wasn't using bad words.
Doctor, is that the same as like a lazy eye or something like that?
No, we're going to talk about lazy eye. You're 2 slides ahead of me.
I see that. I think lazy eye, right?
Well, that means I'm doing a good job that I'm leading your thoughts into the next slide.
Exactly.
Now, here's another problem that happens with children with low muscle tone that have something called nystagmus. This was another word that I had to explain. This usually oscillating movements of the eye back and forth because the eye is not focusing properly. It's searching for the area where it's going to see the best. Now, here's lazy eye, and the technical term is amblyopia. So in the very, very center of the eye is something called the fovea. And the fovea is very, very small, but it gives us our very sharp vision, and it gives us a color vision along with the surrounding area called the macula. Just to give you an idea of the size of the macula, that if I take 10 thumbs and make a circle of the area of the top of my thumb and close one eye, okay, that's the macula. That's all it is. Everything else is peripheral vision. So the macula is that area, and the small area within that is the fovea, and that's where we have our sharp vision. People who have lazy eye are not looking with the fovea. When I can look inside their eye with a certain instrument, and I can see that the fovea is moving all around, or it's fixed and they're looking off center. So those are some of the vision problems that we see with children who have been diagnosed on the spectrum. Children who have the classic autism usually may have strabismus, but they really don't have very many serious vision problems. So I reported this last year, and this was about a child that was going to be diagnosed to be on the spectrum, and we tested him. He had the MTHFR mutation, he had low muscle tone, and because of that, he had poor visual and visual motor skills, and we gave him training with the zone TRAC, which we're going to explain in a minute or two. And we improved his vision motor skills, his concentration, and his academic performance. And after 11 sessions, the diagnosis wasn't even in the mother's mind anymore. So we saved this child a lifetime of bad things happening to him, and no medication. Okay. So we're going to talk now about the zone track. Okay, the zone track was developed from basic NASA research, and it's a goggle like that. And why is it biofeedback? Because it measures how the eye is focusing and immediately gives a sound that's proportional to the focus, so people can learn how to voluntarily control their focus. But as that happens, as the focusing muscle relaxes, it stimulates the brain, and the brain goes into a Zen type meditative state with a big alpha wave, which optimizes all the functions of vision in the eye and the brain. Okay, so this is a video. I think if you click on it, you can't hear it. Yeah. Well, let's see.
This is Dale, and I wanted to show you about a product I purchased called Zone Trap. It's a biofeedback device developed by Dr. Trackman out of the state of Washington. It's a very expensive device. It costs $2,000. I'm out of the U.S. I had to pay extra fees. At FDA license fee, $179. I had to pay the shipping fees, $30. So it cost me $2,230, I think it was. But what it does is it helps to reduce myopia and other refractory errors. And I found it helpful to me. I've had it for 1 month now, and it's helped my eyes to open up more. I'm being able to see more detail. I'm getting my eyesight back. I've been a severe myopic for many years, minus 11 diopters, and I've been doing without glasses for the last I wore glasses full-time for 33 years. Then I took the glasses off 15 years ago, and I've been using the Bates method and relaxation methods and ways to get my eyes to open up and see clear. I get flashes of clear vision usually every day for a few seconds at a time like that. And the Zone Track has helped speed up the process of clearing up and getting my vision more back to normal. So I wanted to show this to you. This is the case for the Zone Track. That's what the case looked like. This is the box that it came in, this size box. And so we open up the case, and the actual Zone Track is a pair of, like a pair of glasses. And this is movable. It's got a hinge there. It's movable. And it just, I'm going to demonstrate this for you so you can kind of see how it works. And this is the control switch. You have two sets of switches. One is for training the left eye and the right eye, and aligning the left eye and the right eye with a little green light. Inside of here, you've got the lights there. I'll describe those to you here in a bit. And you got a battery. This is a Mophie battery pack. It charges. It's a battery charge. So I plug that in. You got a little switch here. And you got a speaker. I got this Bluetooth speaker.
Lying.
So I'm going to turn this on so you can see how it does. And then I'm going to flip the switch here. Okay, I don't know if you can see it, but there's a red light coming out of there. You can see that red light. Can you see it? You got to get it lined up. Hear that chirping? That means it's lined up. See, you hear the pitch of the sound? As it goes higher and higher, it measures the inside of your eyeball, and it connects that with the sound. And as the eyeball opens up wider and wider, letting more light in so you can see clearer, the pitch goes higher and higher. That's how it works. And you do this for about a minute or so, and then you change to the other eye. And you do this back and forth 3 times for a 20-minute session, and you do that every other day. That's the basic training program for the Zone Track. I found it to be quite effective. You hear the sound? It's pretty high pitch right now. And that means my eyeball is pretty well open. So I'm just looking straight into the device, and I have the peripheral vision around the device. So my peripheral vision is taking in more of the eyesight right now. Okay, now I'm going to switch it to the other eye. So I'm going to turn this off.
Okay, so that hopefully gives you some explanation of the Zone Track and how it works. Now, we're going to take a look now at the brain waves. So although there are 4 brain waves, we're only going to talk about 2, the theta wave and the alpha wave. The theta wave is related to poor concentration, and the alpha wave is related to good concentration and the Zen type meditative state. And what you can see here is that before the training, okay, the blue waves are here and here, and then once we start the training, they go off the chart. Okay. Now, for the classic type of autism, we have found that applied behavior analysis, also known as ABA, is the most effective method of training people with the classic autism, and it's used for a variety of problems and to teach a variety of skills. So very basically, the major breakthrough in the treatment of people with autism was in the 1960s with the application of behavior modification known as ABA. Briefly, the method for modifying behavior is based on positive reinforcement, which increases behavior, and negative reinforcement, which decreases a behavior. Now, we have to be careful with the negative reinforcement because it will decrease behavior, but sometimes it will lead to withdrawal and avoidance where the person receiving the negative reinforcement just won't do anything. So we have to be very, very careful. So we emphasize the positive reinforcement and use negative reinforcement only as a last resort. So what type of skills do we teach? Sitting, to get an autistic child to sit, make eye contact. The classic autistic person does not make eye contact. We teach them sign language so they can communicate because they are typically nonverbal. And then we teach them vocalization, and then we can teach them speech. So this is just an example of one patient that I worked with. She was 17 years old, and all she did all day was either sit on the couch and rock or go to a table and drool and then play with her saliva. That was her behaviors almost the whole day. So what did we do? We found out what food she likes to eat, and she understood everything that we said. So the primary positive reinforcement was food, and secondary was praise. If she was non-compliant, we only gave her timeout. And what was timeout? That we would leave the room and just leave her by herself. There was no negative reinforcement necessary. Okay. So after a short time, when prompted, she would get off the couch and come sit at the table. And at the table, she was taught match the sample and the alphabet. And then we started to teach her American Sign Language, and the first sign was more, and she liked that, so she could get more food for reinforcement. And I trained her attendant to be her therapist, and she was finally able to comb her hair and brush her teeth, which she hadn't been able to do before. And then once I finished with the basic program, the attendant was able to continue. So here we took someone who was non-functioning and really changed the life, and also for the whole family. Okay, so this is match the sample. So we start with one color. Actually, we started with black, and then we would have another black square and a circle, and then she would learn how to match those. Then we'd change the colors, and then we would switch the colors, and then we would go to the alphabet. So we just gradually taught her how to make size and shape discriminations. Okay. Now we're going to go back to the hypothalamus, which is one of my favorite subjects, and shows you why the modification of behavior is more than just an outward thing. There's actually things going on in the brain. And so what this shows is that we have two different pathways in the brain. Okay, the green are the non-cognitive pathways, and the blue are the cognitive pathways. And as you can see from all the lines, they're all interconnected. All interconnected. Everything's related to everything else. Sometimes we don't know how the relationship is established or what the connections are, but everything's related. Okay, so now we're going to go back to the original questions. So the first questions are, one, are the changes in the diagnostic criteria due to a broader definition responsible for a higher prevalence? My answer to that is yes. Two, is glutamate, which is related to glyphosate, GMO, MTHFR, is that involved in autism? Yes.
So going back to that, because I asked those questions about those particular parts of the of glutamate, can you explain in a short and concise way how it's related, how glutamate is related to autism?
Glutamate is a brain transmitter. So as we saw that the classic autistic child or person has a higher incidence of synesthesia and also does not respond to the rotating striped drum. So there's something going on in the brain, okay? And we can trace all the recent research back to glutamate, which is one of the major brain transmitters. Okay. The other question is, why is one child on the spectrum and one child not? And we're going to see on the next slide that it's due to different combinations of genetics, and whether both parents have it or only one parent have it. And the last question is there a genetic component? And I say definitely yes. So this is for the MTHFR mutation, which is autosomal recessive. So we all have one set of genes from our mother and one from our father. So in this case, the parents have only one of the MTHFR mutations. And if they had 4 children, one child would have one from each, 2 children would have only one, and one child would not have any. So this way we could see why if you have 2 children or more than 2 children, that your genetic makeup can be very, very different. And there's hundreds and thousands of genes. So the combinations are off the wall. Okay, so we're going to go back to remind us of the 14 different genes related to glutamate. Okay. So once again, I had mentioned, or maybe I didn't mention, that the formula to calculate the combinations and permutations is 2 to the n-1 power, okay? So for the glutamate mutations, n is 14, we take 1 away, so we get 2 to the 13th, which gives us 8,192 possible etiologies of autism just related to glutamate. So now if we include the ones for mercury and we include all the others, it becomes astronomical. And to say that there's just one cause for autism, which everybody's been looking for, it's impossible to determine. It doesn't mean we can't have a successful treatment. It doesn't mean we should stop looking. Maybe there's another common factor. But right now, this leads to all the confusion because some say it's mercury, some say it's this, some say all these different things.
You know, this formula you're talking about, you're saying there's thousands of ways that people can develop autism, right? Like I said, I've got autistic family members. that are on the autistic spectrum disorder or ASD. They're on the ASD anyways. Are you telling me that there's not just one reason that they're on the ASD? Like it's not just a genetic mutation? You know, I mean, if there's 10,000 ways for this to happen, like how on earth do you diagnose in this? How do you cure it? Is there a cure for it?
So that's a good question. And the good answer is that for the MTHFR mutation, we can give supplements. If they have a glutamate mutation and they're lacking glutamate, they can eat foods high in glutamate. Okay. If they have low muscle tone, we can build up their low muscle tone.
But how do you know what's what? How do you know which one is which?
You do it systematically. You see if they have low muscle tone. If they have low muscle tone, you build their muscle tone. If they have you can do genetic testing and see what mutations they have. Once again, go back to the epigenetics video on the YouTube channel. So we can address all these different things. You don't always have to know the exact cause to provide a good treatment.
But what you're saying is there's not. But what you're saying is it's not like a magic bullet. There's not a magic solution for any particular person. It depends on, you know, identifying what their person's root cause is, where it's coming from, and then and hopefully you figure out what that root cause is. So then you can plug in what's missing or whatever else. So you can figure that out. Is that right?
Yes, and that's so. Over the years, I've become an empiricist, and although I'm interested in the theoretical concepts involved in these things, the most important thing is to make people function better. So the treatments that I do, the biofeedback and the methylation, have no bad side effects. So they either work or they don't work. And in most cases, like 90% of the time, there's substantial improvements. Okay. Now, what's the bottom line here? What's the advice? A, have the genetic testing done. B, if there's hypotonia, vision, physical, and occupational therapy are recommended. C, avoid glyphosate and foods with GMO. D, eat healthy foods and drink lots of water. E, be careful about vaccines with mercury, and go to doctor hunting and avoid labeling. So if you go to a doctor and the doctor's adamant in saying your child has this or your child has that, and the tests are soft tests. What do I mean soft tests? That it's not like taking a temperature. You can go to the doctor who'll take your temperature. He can measure your heart rate. These are hard signs. There's no hard signs for these diagnoses. So go doctor shopping find a doctor that tells you what you want to hear. Okay, so going back, so 2 to the n minus 1 to the 11th is 2048.
What does that mean?
Different possible combinations based on that diagram. Okay, and what's the summary? Two definitions of autism, classic and ASD. For classic autism, we have ABA. For ASD, we have genetic testing, epigenetic, supplementation, vision therapy, physical and occupational therapy. And although the cause for autism remains elusive, we're still learning more about genetics from the Genome Project. And lastly, from what our current knowledge is, glutamate appears to be the most common link among the many etiological factors of autism, whether classic or spectrum.
So I have a quick question for you. I understand you're a doctor of optometry, right? You also have your PhD in experimental psychology, right? So I'm looking at it from this perspective, from that intersection, all right? I'm wondering, from your opinion, if you think that autism, a classic or autism spectrum disorder, either way, is it more of a function of ocular, or is it more of a function of psychological stuff, or is it, or like where in general, where is autism coming from? Is it ocular, psychological? What's going on?
Okay. So do you remember the diagram of the hypothalamus?
Sure.
That's where most of the action's going on.
But is that like more ocular? Like what
It's everything. It's every hypothalamus.
chemical imbalance?
The hypothalamus regulates everything, and in biology and psychology 101, we learn that the body wants to be in a homeostatic balance, not too excited and not too schleppy. I'm from Brooklyn, so we could use that word. So anything that disturbs that balance is stressful and causes either chemicals not to work right or nervous pathways not to work right. Something goes wrong. And now that we throw genetics into it, it adds another level of complication and the current confusion. But nevertheless, we still have effective treatments and gave you the recommendations.
Excellent. Thank you. I appreciate that. One more question that I have for you. Okay. So really quick. So if sat through your presentation, right? I spent the time to understand this. If, say, I have an autistic child or sibling or friend or anything like that, if I think that somebody's on the autistic spectrum in general, what should I do for them, and how can I possibly help them without being too without being like offensive or too aggressive or anything like that?
Okay, so you can send them to my YouTube channel, and that will answer some of the questions. And the other is to explain that there's many factors involved: genetics, low muscle tone, vision problems, all these things, and they should be systematically examined and see where the problem area or the problem areas are. Are there any other questions?
So you were talking about making sure that a doctor never labels you. So I have a question. So I was diagnosed with ADHD, but that I went through a soft test to get that medicine. So would you say that ADHD is not real? Or like, talk to me on.
This is a good question. So once again, we go to the difference between hard signs and soft signs. Okay, there are no hard signs for ADD. It's all questions. And this is something that I did. Before there was ADD, that problem was known as minimal brain damage. Okay. Around the 1970s, they changed it to ADD. I guess parents didn't like their children being called minimum brain damaged. For minimal brain damage, there was a physician's manual on how to make the diagnosis. In the beginning part of the manual, it says if there's a vision disorder, a hearing disorder, or a neurological disorder, you cannot make the diagnosis. All the children that I have seen who've been diagnosed ADD have a vision problem. They should never have had that diagnosis made. It's terrible. That's number one. Number two, going through the manual, I found 100 questions. So when I was teaching at the university, the first class, I would hand out a piece of paper that had 4 columns, 1 to 25, 26 to 50 with a little box next to it, and I'd read them the 100 questions. Okay, out of hundreds and hundreds of students, I only had one who hadn't been diagnosed ADD. It's meaningless. And it's terrible to label someone who has, for example, a double vision problem. To say that they have ADD has nothing to do with that. And you could give the test over and over again, and you're going to get different check marks on the boxes. That's not science. Who wrote the manual for MBD? Shibagagi. Shibagagi makes Ritalin. I think that answers your question. Okay. Any other questions? Okay. So I'd like to thank you for coming, and especially thank Brian for his audiovisual expertise. Have a good night.